Okay, everybody, we'll probably get started now. So I've already said it, but hi, nice to kind of meet you as it is. Sorry. My name's Alex, and I'm going to be teaching you today about cell structure and functions at the GCSE level. So hopefully you'll understand, but if not, if you have any questions, obviously feel free to put those forward at the end. So before we begin, I just want to give you a bit of background to my education. Hopefully, give you an idea of what you can do after studying biology at GCSE. So for my actual school, I studied in a state school in Kent from 2011 to 2018, in which time I took my GCSEs and my A-levels. So in my GCSEs, I was lucky enough to get 12 A stars and an A, and you can see me here on the left, very happy on results day. And then I went on to complete my A-levels, where I studied biology, psychology, Spanish and maths. And I also got an A star at biology A-level, as well as psychology. And I'm hoping, obviously, that means I'll be a good person to be teaching you biology today. So what did I do after that? Well, after my A-levels, I went to university. So currently I study at the University of Cambridge. So I'm at St. Catherine's College, which if you're considering applying to anywhere, definitely would recommend applying here. And so I've just finished my second year studying psychological and behavioural science, which is basically psychology, biology, and a bit of something else. So that's really fun. Once again, has allowed me to keep developing my biological skill set, which is of course really important for me, but also important for me teaching you today. So more specifically, during my degree, I studied education, evolution, neuroscience, and like social psychology and stuff like that. So yeah, definitely a very interesting and varied course. But enough about me. We can start talking now about cells, as that is the topic of today's lesson. So all about cells. We'll be focusing on animal and plant cells today. So to begin with, we're going to focus on some general facts about cells. So all organisms are made up of cells. Most organisms are multicellular, meaning they're composed of numerous types of cells. However, most of them do have cells that are specialised to do a particular job. Microscopes are needed to study cells in detail. So what this means is if we want to really understand what's going on inside the cells, we need to use something called a microscope. Now, I'm sure you've probably encountered microscopes before, but nonetheless, we will definitely go over that today. So different microscopes. So maybe you're not aware of this, because I know sometimes in school we get given the same standard microscope, and that's as far as our scientific understanding goes. But there are actually different types of microscopes, and to study cells, we can get kind of a range of features from using each microscope. So the human eye can only see objects as around kind of 0.05 millimeters, which is obviously tiny. So a microscope is required to see any detail in these cells. So what microscopes actually do, and the way that they work, is they magnify an image, meaning it appears larger. Obviously, if it appears larger, it's a lot easier for us to see it, to see the cell, and therefore we can kind of understand what's in a cell, how a cell works, from being able to see it. So the thing that we specifically need a microscope to see is something called organelles. So organelles, this is a really important term that you're going to definitely need to use, especially in terms of in your exam, when you want to be hitting those key phrases, keywords. The word organelle is very important. So an organelle refers to a smaller structure within a cell, and we will come to some examples of this and what they are. So like I've said, to view these organelles, we must use a microscope. But the microscope we're focusing on here is something called an electron microscope. So an electron microscope is actually a tool that can be used to give us almost as in detail view of a cell or of a sample that we're viewing as possible. So this is in comparison to something called a light microscope, which is an alternative type of microscope. So the difference with this is rather than using a beam of electrons to help view the sample, we just simply use light on it and magnify the image like that. And whilst this still has its benefits, obviously, the light microscope does not give us as much of a detailed image. It doesn't give us as high resolution. So that's another key phrase there. A light microscope does not have as high resolution as an electron microscope. So we can use the light microscope to identify the outline of a cell, so where the cell is, and its largest organelles, for example, the nucleus. But if we want the real detail and to be able to see all of the organelles present in the cell, we really need to use an electron microscope. So we will now move on to look at the diagrams of different types of cells. We will focus on labelling these diagrams and recognising their structures, and then we will move on to the function of each. So the reason that this is so important is because any exam question that you get, especially at this level, regarding what we're going to cover today, is often about diagrams. So whilst there are many questions you can get about cells, for example, cells in immunity, specialised cells, cells that photosynthesize, for example, the point of today's lesson is to really be able to identify the structures within a cell and then say what their functions are. So most commonly the question will literally ask you to label a diagram, and that's why we're going to spend so much time looking at this. So we'll begin by looking at animal cells. So and in this picture, I've got two diagrams of an animal cell, and I want you to note the difference in detail provided by each microscope. So on the left, we have what you can see from an animal cell under a light microscope. So the exact same animal cell, these are not different cells. It's just on the left, what we can see with a light microscope, and on the right, what you can see with an electron microscope. And this obviously gives you some insight into which microscope you'd want to use to study cells and why I mentioned that previously. So we can see here that an animal cell consists of cytoplasm, the nucleus, ribosomes, the mitochondrium, and cell membrane. Now with a light microscope, we can only obviously see the cell membrane and the nucleus. The cytoplasm is something we can assume is there, we don't actually have to see it. And in the electron microscope, we can see all of the aforementioned organelles. So key things to remember here, not your scientific definitions, because we will get onto those, but in terms of how can I spot these on a diagram? Firstly, practice, 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 which we will get to. But secondly, some kind of key tips to help you. So the nucleus is always the largest, darkest blob on the page. Once again, reinforcing here that this is not the most scientific way, but it's just something to help you remember it. So mitochondria will typically be drawn, if I could just get a pen, sorry. A mitochondria will typically be drawn in terms of an exam paper, either like it is here, so we can see it's kind of an oval with like a fishbone structure in it, or if you're asked to draw it yourself, you can draw it like this. And this is kind of the accepted way to present a mitochondria. So just a little tip there, if you see that on a diagram or if you're asked to draw it, that's how you draw a mitochondria. A cell membrane is not hard to spot, it's literally the thing on the outside. Ribosomes may appear as little dots, specifically clustered together. Yeah, there you go. That's your kind of tips for drawing. So a quick fact about animal cells, they range between 0.01 millimeters and 0.05 millimeters in size. Definitely important to remember this, especially when we consider what the range of the human eye can actually see. It's very important to then be able to compare this to animal cells. So make a note of that if you haven't already. Cool. So now we're going to move on to really considering what each of the aforementioned structures actually has, what it actually does to the cell. So first off, we'll begin with cytoplasm. So the cytoplasm is a jelly-like material that contains dissolved nutrients and salts and structures called organelles. So dissolved actually can be defined as becoming incorporated into a liquid. So what this means is nutrients that we need are present in this jelly-like substance, which we find in every animal cell. Also, the organelles that we talked about previously, so your mitochondria, your ribosomes, your nuclei, everything like that can also be found in this jelly-like substance. And because of that, that's why when we label the cytoplasm, we can literally draw a line to anywhere in the cell, as long as it's obviously not directly touching one of the other organelles, because the cytoplasm spans the whole cell. So this is actually where many of the chemical reactions that the cell will carry out. This is where they take place, mainly because obviously they contain the nutrients needed for many of these reactions. It's also a transport medium. So what a transport medium means is, for example, the blood is also a transport medium. It means that it's a way that we can transport or move nutrients and important substances around the body. So yes, everybody have a quick look at the diagram. So key here to be able to define what cytoplasm is and be able to draw cytoplasm, in which case you just draw the cell and label it. So I'll just give you a second to really take that in, and then we'll move on to something else. Cool, so next up is the nucleus. So the nucleus stores genetic material, specifically DNA, which controls the cell's activities. So this DNA is really responsible for the cell's activities. And I think within your GCSE, you'll definitely move on more and focus more deeply on what DNA actually does and why it's so important. Obviously, that's not the purpose of today's lesson. However, just to emphasize, the nucleus really is kind of the mind of the cell. Now, I wouldn't use that definition personally. I would say the nucleus stores genetic material, as I've written here. But just to give you an insight, the DNA which this nucleus stores is responsible for all of the coding, and therefore the replication of all of these cells, as well as how the cells function. This is all coded for by DNA. And that's obviously why the nucleus is such an important organelle. So you can spot this very easily on a diagram, as it's always the largest, kind of darkest circular shaped component of the animal cell, as I've already mentioned. So definitely worth remembering that. Also to make you aware, obviously normally on your typical diagrams, they look like this. So this is kind of your typical diagram that you'll get in a GCSE question, or one like this. However, you need to be familiar with looking at more complicated diagrams. For example, this diagram here, the nucleus is not this small purple inside, it's this big kind of, sorry, that was an appalling circle. It's this big ring on the outside of that, circled by the green in this case. And obviously that's not our standard representation of a nucleus, so we have to kind of think outside the box there. Also, if given a 3D diagram, it is definitely more difficult to kind of assess what's what. But once again, we should be able to tell here that this is the nucleus because it's kind of a large circular shaped or spherical shaped object. So once again, I'll emphasize always practicing. It is difficult to find questions that look like this on the GCSE past papers because they are quite infrequent, but that doesn't mean we can't look online and practice those. So next up is the cell membrane. So unlike plant cells, which we will cover next, animal cells do not have cell walls. Instead, they only have cell membranes. So this is really easy to spot because it's just going to be on the outside of the cell. So animal cells do not have a cell wall. They do not have anything else surrounding them. So the cell membrane is simply the thing on the outside, making it very easy for you to label. So key bit here to remember that animal cells do not have cell walls. So cell membranes are permeable to some substances, but not to others. So permeable means it allows liquids or gases to pass through. And obviously that means, for example, things like oxygen can enter the cell, things like carbon dioxide can leave the cell. However, certain substances wouldn't have the capacity to enter, which is obviously kind of a protection mechanism of the cell. So therefore, because of this permeability, the cell membrane is actually responsible for controlling the movement of substances in and out of the cell. So that's obviously a very important point there. I'll just give you some time to jot down any notes you might be making. But yeah, it shouldn't be too difficult to label. It's always the most outer kind of layer of the animal cell. So next up we have the mitochondria. So the mitochondria are really important. On the left you can see, hi to whoever just joined. On the left we can see some diagrams of mitochondria. So obviously, as previously mentioned, you can definitely, if drawing yourself, draw them with this kind of S-shaped squiggle. And that works perfectly. And just be familiar of what they look like so that you can actually recognize them on a diagram of your that you may be given in the exam. So we've got kind of a more detailed example down here at the bottom, which we can see smaller scale up here, kind of resembling this squiggle shape that I've drawn down below. So what actually are the mitochondria? So these organelles contain the enzymes that we use for respiration. And obviously we know how important respiration is. It is essential for living organisms. So this is where most energy is released in respiration. So it's really all kind of centered predominantly in the mitochondria, not completely, but predominantly. And a typical phrase that you may have heard before is the mitochondria is the powerhouse of the cell. Now, I think you can definitely one up your answer by, instead of that, you can definitely say, you know, these organelles contain the enzymes that we use for respiration. This is where the majority of the energy is released in respiration. But if not, when in doubt, the mitochondria is the powerhouse of the cell. So yeah, just give you a second to jot down any notes you may have. Just make a note of the structure for if you're drawing your own. Cool, so next up we have ribosomes. So ribosomes are tiny structures where protein synthesis occurs. So that's really important. So ribosomes are where protein synthesis occurs. So protein synthesis, if you haven't guessed it, is the process in which proteins are made or synthesized. So if I said to you, for example, I know I wouldn't, but if I said we're going to synthesize a cake, that means we're going to make a cake. Doesn't really make sense. But the point here is protein synthesis means making proteins. So a more detailed answer, if you want to really kind of hit those high level marks, you can say, when it says, what is the function of a ribosome? So a ribosome's function is protein synthesis, as it joins amino acids together in a specific order. So that's really what protein synthesis is. So whilst this is another topic that I don't want to cover very deep in today's lesson, it's important to remember that protein synthesis involves joining amino acids together to form a polypeptide chain. So that's joining amino acids together to form a polypeptide chain, which can then be used to create a protein. So these ribosomes are the actual things that are responsible for doing that. They hold the amino acids in place during this process. So definitely very important. On a diagram, as we already mentioned, a lot of the time you can present your ribosomes as small dots. They'll definitely be clustered together and should be quite easy to spot on a diagram, but just to give you a bit of insight, this is what they look like close up. So you can see from this picture how come they are presented as dots, obviously on the diagram, some very small kind of circular shaped organelles. Okay, so time for a quick recap. Obviously, I can't see your answers, but just have a go for me. I'm going to ask some questions, very simple questions. Just write down your answers and don't let me know, obviously, but just see how you get on. It doesn't matter, like you've only just learned this today. Just give it your best. So firstly, hopefully you can all remember this one. What is the function of a ribosome? So is it A, to release energy from glucose? Is it B, for protein synthesis? Or is it C, to build sugars by photosynthesis? So just give you a bit of time. A, B, or C. So hopefully you got the correct answer of B. The ribosome's function is protein synthesis, as we already mentioned. Cool, so moving on. To view small organelles, which type of microscope must we use? So to view the small organelles, which type of microscope must we use? Bonus point if you can write down why. Yep, so hopefully you got that. To view the smallest organelles, we must use an electron microscope. Now, the reason that we have to use a microscope in general, as we've already mentioned, is because the human eye can only see a very small kind of range of images, and obviously we want to magnify an image to make it accessible to humans. Specifically an electron microscope because it can give us a more detailed image. It has a higher resolution than a light microscope, allowing us to see the smaller organelles. Okay, final question. Where is genetic information stored in the cell? A, the mitochondria, B, the cytoplasm, or C, the nucleus? So the answer is C. So genetic information, specifically DNA, is stored in the nucleus. So hopefully that's all good for you. So now we'll be moving on to plant cells. So plant cells have so many similarities and they share many features and organelles as animal cells. However, there are also many differences, so we'll definitely discuss that today. So one key difference is that a plant cell size ranges from 0.01 millimeters to 0.1 millimeters. So hopefully you can remember how that differs to the size range of an animal cell. So now we're going to look at the basic structure of a plant cell. So this is your typical plant cell diagram. So I want you to have a think about which organelles a plant cell has and an animal cell does not. So I want you to have a quick look at this diagram and start thinking to yourself, okay, which of these organelles does a plant cell have that an animal cell does not? I'll give you a little bit to think about that. Cool, so we can see from this diagram here, we've got cytoplasm, which is, you know, wherever the cytoplasm is, whatever the cell it's in, cytoplasm is still cytoplasm. It's still that jelly-like substance. which has dissolved nutrients in it and also has the organelles in it. So we can see that obviously a plant cell has cytoplasm, and we know that an animal cell also had cytoplasm. Next one down is the nucleus. So once again, all cells will have a nuclei, or all plant and animal cells will have a nuclei, apart from a few exceptions, which are specialized cells, and this controls and holds the DNA of the cell. So next up we have ribosomes. Ribosomes, once again, as I already said, they can be drawn as little dots on the diagram, and they're often clustered together. And so we can see here that plant cells have ribosomes, but obviously we already know animal cells do too. Now, next up is the vacuole. So obviously we will come to this in greater detail, but we can see here our vacuole is this large kind of shape here. So whilst we don't yet need to worry about what this is, it's interesting to see that plant cells do have this, whereas animal cells did not. Next up, mitochondrion. Yet, once again, we already know both animal and plant cells have that. Cell membrane can confirm that they both have that. And then we get to the cell wall. So as I already mentioned, plant cells have a cell wall, whereas animal cells do not have a cell wall. That's one of the key differences that we should be considering here. So a thing that people always get wrong, so a place that you can definitely pick up marks on an exam question, is avoiding the confusion between a cell wall and a cell membrane. So obviously, the cell wall is the most outer part of a plant cell. The cell wall is not the most outer part of an animal cell, because animal cells do not have a cell wall. And once again, this diagram just really helps you to see the difference between what you can see with a light microscope versus an electron microscope. So hopefully you've all got a good look at that. If not, I'm sure you get sent these at the end of the lesson, but alternatively, you can just kind of search into Google diagram of a plant cell, and it should come up with something very similar to this. So as we've already mentioned, the features here, or the organelles here, that are specific to only plant cells are the vacuole, the cell wall, and the chloroplasts. So what are chloroplasts? So they kind of look like this. So you'll see them as small kind of oval structures, and this is what they look like on the inside. And so organelles that contain a green pigment called chlorophyll. So that's really important. So if you're asked what are chloroplasts, the first part of your answer should be they are organisms containing a green pigment called chlorophyll. Now, chlorophyll is important as it actually absorbs light energy for photosynthesis. And we know how important photosynthesis is for, obviously, plant survival and function. So chloroplasts contain the enzymes needed for photosynthesis. On the diagram, chloroplasts will typically be drawn in green. So I think we can see that in the last, chloroplasts are drawn in green here. However, obviously in your exam question, you probably have them in black and white, which obviously makes it a lot harder to identify. So they will appear as dark ovals, and they're smaller than the nucleus, but larger than the ribosomes. So you shouldn't be confusing, they'll be similarly sized to mitochondria, as we can see here, but you shouldn't be confusing them with the mitochondria, because as we've already mentioned, those mitochondria will have your kind of, what I like to call the fishbone structure, or the S shape going through them. So next up we have the cell wall. So another really important thing here, cell walls are made from cellulose fibres, so cellulose. The cell wall actually strengthens the cell overall, which therefore supports the plant. So if you think, each cell wall gives support to an individual cell, but when these are accumulated together in a plant, that supports the total plant. So the cell wall is always the most outer aspect of a diagram, like I've mentioned, as long as it is a plant cell. If it's an animal cell, there is no cell wall at all. So please remember the difference between a cell wall and a cell membrane. Okay, last one is a permanent vacuole. So we call it a permanent vacuole. You can just call it a vacuole. And basically this is a big, I like to think of it, vacuole, big hole, and it's filled with cell sap. So what the cell sap does is it helps to keep this, the cell turgid. And what turgid means is swollen or filled up, which obviously helps to give the plant structure. So if it wasn't turgid and it wasn't kind of rigid like that, the plant would then wilt. So whilst keeping it turgid may not sound like a positive thing, it really is. It should normally be, it's quite easy to spot on a diagram. So as we have here, it's a very large part of the diagram, and it looks a bit like a lake. Okay, so time for another quick check-in to just see how you're getting on. What are cell walls made of? So what are cell walls made of? So have a go at that. Yeah, hopefully you all got that right. So cellulose, you'd have also been fine if you said cellulose fibres. Either way is correct. So well done. So on average, what size are plant cells? So on average, what size are plant cells? Don't forget to give me your units here as well as just a number. Obviously, otherwise you won't be correct. So the answer is 0.01 millimetres to 0.1 millimetres. So hopefully you all got that and can compare that to the size of an animal cell. Okay, well done everybody. So now we're going to move on to exam questions. So I understand obviously that we're moving quite fast here, but that's because in my opinion, the key bit here is to practice, practice, practice. So once you've learnt your definitions, after that point, it's all about practicing labelling and interpreting diagrams. So for example, a question could say to you, what is the function of part A? Now it will be a diagram with part A labelled, and in that point you have to firstly identify what part A is and then remember part A's function. So it's really important to be able to do both aspects here. So as I've kind of mentioned, exam questions relating to what we have covered today are often quite simple. They're never too complicated. So you'll be frequently asked to label a diagram or perhaps even draw your own diagram representing the defined parts of a cell. For example, if I said draw an animal cell, on your diagram represent the mitochondria and the nucleus, that's what you would do. And so you should practice a variety of questions in order to get used to the style and shapes presented to you. So what I mean by this is that every diagram will vary a little bit in how everything's presented. For example, on one diagram, your mitochondria may just be presented like this, as I've mentioned. Sorry, that's a poor drawing. Or on another diagram, it may be kind of in a lot more detail like this. That still doesn't look very high detail, but hopefully you get my point here about the distinctions that can be made between each. So definitely make sure you're able to recognise each part of the cell in black and white. So don't rely on purely recognising the chloroplasts because they're green, because in your exam, sadly, they probably won't be. So question 1, I'll give you some time to go over this. So question 1, part A, label the nucleus and the cell membrane in figure 1. So what this question would be requiring you to do, so obviously you don't have the question in front of you, but you can quickly draw out something at home that looks like this. This is what you've been given so far. They told you that that's cytoplasm. So it wants you to label the nucleus and the cell membrane. So I'll just give you a bit of time to have a go at that, and then I'll show you what I think. Yeah, cool. So we should have here that the nucleus is this part here, and unlike lots of diagrams, the nucleus isn't particularly dark or particularly large here. However, it is definitely the largest feature of this, the largest organelle present. So we can definitely kind of assume that that's the nucleus. So I'm just going to put N because it's hard to write on the screen, but that's the nucleus. And then your cell membrane should be this outer part here. So you can literally label it. So I would personally write cell membrane in your exam. But you could literally label it here, 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 like the cell membrane spans the cell, so that's fine. So hopefully you all got that, and that will give you your 2 marks. So I didn't put part B in because it just wasn't relevant to what we've covered today, but moving on to part C for 1 mark. Ribosomes and mitochondria are not shown in figure 1. What type of microscope is needed to see ribosomes and mitochondria? So hopefully you all wrote down electron microscope. So we've already mentioned quite a few times that an electron microscope is the kind of tool that we need to have a look at cells in detail. Now we already know that the mitochondria and ribosomes are small organelles, specifically the ribosomes, they're very tiny. So it's not surprising that we can't see them on this diagram. And as I've already mentioned, obviously I couldn't say it before because you hadn't answered the question, but we can see here that, like I said, the nucleus is quite small relatively. So perhaps we can conclude from this that we've used a light microscope here, and that's why we cannot see any other organelles on the diagram. So yeah, an answer to this one is electron. I'm just going to put elec for the purposes of writing. Next question, question 5, which is actually question 2, so don't worry. The diagram shows a cell. So the cell you can see here is animal or plant. I'll leave that to you to decide. So always have a look at the key. It may or may not be relevant to you in the question, but always make sure you've looked at the total question. So use the words from the box. So this is giving you a hint to label the structures A and B. So what is A? What is B? Cell membrane, chloroplast, cytoplasm, or nucleus? So I'll give you a bit of time for that. Okay, cool. So hopefully what you've all got here is that A is the nucleus. So, you know, this is a large, dark, circular shaped structure or organelle. So that's the nucleus. So I'm going to put this as A. Once again, in your exam, please, if it says right on the line, right on the line. And then B, so what could B be? I'm going to say cell membrane. And that is your correct answer because this is the most outer part of the cell. So you should be noticing at this point that this is an animal cell, not a plant cell. And you can get that just from the kind of general shape that you're presented with. So not all animal cells and not all plant cells follow the circular and kind of rectangular shape that we've kind of covered today, but typically in this type of question they will. So the cell in the diagram is an animal cell. How can you tell that it is an animal cell and not a plant cell? Give 2 reasons. So 2 reasons why you know that this is an animal cell and not a plant cell. So I'm going to go back for you and so you can have a look at it. I'll keep flicking back and forward between the 2 pages so you can have a look at answering that question. Okay, cool. So last bit of time. How can you tell it's an animal cell and not a plant cell? Give 2 reasons. So in this question, what we're looking for is basically it's asking you, do you know the difference between an animal and a plant cell? And yes, hopefully you do. So we've already discussed how a plant cell should have things that an animal cell doesn't have, such as a vacuole, a cell wall, and chloroplasts. Now that's 3 reasons I've given you there, so you can break that down and choose 2 of those. So, you know, I can tell that this is not a plant cell because, for example, there are no chloroplasts, there is no cell wall. So we already know that everything an animal cell has, a plant cell also has. So, for example, you wouldn't be able to say.
I think it would be. Wait, does every plant cell vacuole?
So basically, the plant cells so we have different types of plant cell. So we have specialized plant cells, in which they may not have a vacuole. But for the purposes of this lesson, so when you'll be labeling your basic animal and plant cell diagrams, you will always be drawing a plant cell with a vacuole. So when we get onto covering specialized cells, then you'll realize that they do differ. For example, not every single animal cell has a nucleus, which is kind of a crazy idea, and not every single plant cell will have a vacuole. But for the purposes of this, as you can see from the exam questions we've been going over, they will all have a vacuole. So as I was saying, so 2 reasons why this is an animal cell, not a plant cell. You just literally need to choose out of what we've already said. There is no cell wall present. There is no chloroplast present. There is no vacuole present. Okay, cool. Moving on to the last question quickly. So question 7. So the image below, I'm not sure if you can see the top of that, but the image below is basically showing us a cell here. Use the words from the box to name the structures A and B. So this is obviously a plant cell. We should be kind of guessing this from the general shape. So if you could label A and B. Actually, I'm not sure if I'm on the right page. God. Sorry, my laptop has frozen, but I'm not sure if you can see the plant cell question or potentially you can see the stomach question. So I'm not sure if anybody's able to unmute me and let me know if you can hopefully see something. But
we can see this. We can.
Oh, I'm so sorry. Thank you for letting me know. It's sudden you cut out just as you're about to tell me, but thank you. I appreciate that. Okay, so hopefully now you should be on the stomach cell page. I'm hoping you can see my screen. It's having a bit of a freak out. But either way, if you could label the structures A and B for me. And when you've got your answer, we'll go over them. So on this slide, we can say that A is definitely the cell membrane, because it tells you here that this is a stomach cell. So it tells you that this is a stomach cell, so we know that there will be no cell wall present. And then we can see that B is not referring to the nucleus, so that's a common mistake. People will just see this kind of here and go with that. But B is actually pointing to what we'd view as nothing, which means we conclude that B must be the cytoplasm. Then final question, what is the function of the nucleus? Tick one box. So is it to control the activities of the cell, to control movement of substances into and out of the cell, or to release energy in respiration? So hopefully you've got here that the answer is to control the activities of the cell, and it obviously does this because DNA is what controls it and the nucleus houses this DNA. And so I believe that that is the end of today's lesson, but obviously as I've got kind of on this slide, it will just show to you how many questions are out there. So there are so many questions that you can find online on lots of different sites. Questions by topic are really good for finding ones that are specifically about this. And like I say, keep practicing is definitely, keeping to practice it will definitely help you in this topic. So does anybody have any questions? So feel free to enter them in a chat, or alternatively, you can say them out loud if that's possible. No, so nobody has any questions? Cool. Well, that's totally fine with me then, as long as everybody understood that. Yeah, okay, cool. So if you are keen to have any further lessons, obviously, just as it says on the slides, contact Chan, yeah, regarding some tuition with Libra education. But for now, that's all from me. So thank you very much for paying attention. Hopefully that helped you. Yeah, have a lovely weekend. Bye, everyone.